Thank you. I'm not sure this is working because I can't hear any echo. It is? Okay. Good. Let me make sure it's connected. Great. So thank you for that kind introduction and all those memories going down memory lane. I'm going to talk about a topic which is very close to many things that we do in bioinformatics and in informatics in general, socio-informatics and medical informatics, informatics involving any type of search or mining for pattern comes under this category, which we're calling large-scale correlation mining. So you're mining for correlations every time you do a Google search. Right? You put in a text, or you maybe go to their image search facility. You're trying to find correlations between the query and the database. And anytime you blast or go to gather or any of the ontological search sites that we use in order to find out more about the biomarkers that come up. And most importantly, what I'm going to be talking about here in terms of the examples is when you're doing very high throughput biomarker collection, where you're collecting samples on perhaps a human genome or some other very high dimensional quantity, and you're searching for patterns that might exist in the interactions between those variables, not in the average levels under treatment A and treatment B. You know, are the average levels different? But rather the interactions between the variables, which may be completely buried under noise in the mean levels. So that's what this is all about. And so what I'm going to be doing, since I got this very, I think, cogent introduction from Brian on mathematics, there will be some mathematics here, and I hope when you leave this room you'll be convinced that mathematics is key to being able to describe just how many data points you need, how many samples you need, what kind of correlations can be discovered in a data set. If you are limited in the number of samples you have and you know how many variables are in the data base, which might be, you know, an RNA-seq chip, for example. And so that's going to come up. There's going to be some mathematics. It's not going to be heavy emphasis, but it's going to be there. And I hope you'll appreciate how useful it is by the end of the talk. So I'm going to give some motivation and go through principles, basically the mathematical principles underlying correlation mining that leads to tools. Tools that can be used to describe whether you have sufficient amount of data to make a statistically and perhaps biologically significant finding on correlation. And then we're going to talk about a couple of applications, which one being network analysis, network discovery, descriptive extraction of correlations from very large sets of biomarkers with few samples, sample-starved type of framework. And then another application which involves prediction, where instead of trying to correlate biomarkers together from the same class, you're trying to correlate one set of biomarkers to another set. And we'll see that the theory that I developed here really nails this problem. Okay, so first, some acknowledgments of some of my collaborators. First and foremost, Bala Rajaratnam, who I've collaborated on the mathematics that I'll be presenting here, and Hamid Farouzi, who's a current student, PhD student of mine in ECS. Rob Brown and Yongshen Wang were early bioinformatics students of mine. Rob Brown applied here, got in, I believe, to the bioinformatics program, but decided to go to UCLA. He preferred, yeah, he preferred the weather out there, perhaps. Yeah, especially today. And then Jeff Ginsburg, Amy Zass, and Chris Woods at Duke Medicine who provided the data that I'll be presenting here. And then many sponsors. All right, so let's go to motivation. So why are we interested in correlation? What can correlation yield in terms of insights that just looking at average difference between two treatments can't reveal? Well, I have two pictures here. This comes out of a data collection that we designed with our Duke collaborators, and I'll be talking more about the specifics of this data collection. I'm not going to describe what this is other than to say you have blue and green dots on a plane. The plane has, it measures the expression in terms of abundance of mRNA from an Affymetrix human 133A chip for two pairs of genes. The colors on these dots, which denote different samples, are two treatments. They're actually not treatments. They're effects, two different effects from inoculation with a virus. The blue are those people that got sick, and the green are the people that did not get sick, that were able to control the virus and exhibited no symptoms. And solid are women, hollow correspond to men, and then the size of these dots is the hours elapsed since they got inoculated with the virus. So what I'm trying to show here is that if you look at the means, which are in these two diamonds here of the blue and green treatment sets, they're very close together, right? But look at this. These are the regression lines, the correlation lines. We have effectively a sign flip. Positive correlation over treatment 2 between these two genes. They're both co-expressing in the same direction. Sometimes positive, sometimes negative. In some people, it might be they're both positive, and some others, they might be both negative in that particular treatment class. And the other treatment, it's a negative correlation. So this corresponds to a very significant contrast, which is not revealed in the difference of means. The difference of means is on the order of, 0.1 fold change, 0.1, 0.2 fold change. So correlation is the key here in terms of these two genes. Now this is another example where we also get a similar type of conclusion where correlation is actually serving as a proxy for picking out changes in treatment due to an average change. So we have two clusters here. These are males, these are females. And this is a Y chromosome gene that separates them. Okay, so you see that separation through, again, this type of correlation difference. All right, so this I hope motivates why we're interested in correlation. We're also, of course, interested in the mean, but for the purpose of this talk, I'm not going to be talking much about the mean difference. I'm going to be focusing on these correlations that we're going to try to extract. All right, so what is correlation mining? Correlation mining is taking data which is very high dimension, like mRNA expression, or maybe various types of symptoms under different conditions in a respiratory viral challenge study with different viruses, or maybe activation maps if you're doing a functional MRI study of particular types of connectome differences that occur under 2 or 3 different treatments. Or maybe you're looking at tissue engineering, where you're trying to, based on, say, the electron backscatter diffraction data, come up with a way to combine 2 different types of microscope measurements of your particular sample and correlate them in order to figure out what the geometry of the, say, the fine grains in this calcium synthetic bone sample formation processes are. So you take this data, you sample it with some budget. This is going to come in the future in the example, the Spark example. You have a budget. You can't purchase an arbitrary number of samples. They're very expensive. So there's a budget that determines what you sample, where you sample, and how you sample. And there's a value of a particular form of information for the particular type of inference you're trying to do. And then you extract features, you do your correlation mining to find correlations between features that could be different, associated with different genes or different symptoms, different locations in a functional MRI connectome study. And then you form visualizations or inferences, gene pathways where each one of these corresponds to a gene. Each node in this network is a gene. The edges correspond to the amount of correlation that exists between those genes. So if they're very close together, it means they're highly correlated. And then you can do clustering or correlation to find patterns in the connectome that are spatially organized that lead to cognitive pathway discovery or discovery of difference between ADHD and control or what have you, right? So that's the key to making this work is error control. Right? We have an enormous amount of data we can work with, but we all know that data alone is not sufficient to make inferences. You can have too much data, right? There's the principle that the more data you have, the better you are, is false. Unless you have a good model for how that data dimension can be fit into some kind of lower dimensional description of the mechanism that generated your data, having more data is actually a curse, not a blessing. Because you effectively start to find spurious correlations everywhere. And that's where error control comes in, and that's where the mathematics is going to come in finding ways to control that error from the outset without knowing what the true correlation is, only knowing what the number of samples are, how many variables you have, that is biomarkers you have in your set of genes, let's say, and the desired level of correlation that you're looking for. The lower the correlation you're looking for, the more samples you're going to need, right? If you have a correlation equal to 1, you can pick that out with 2 or 3 samples. But if lower correlation, you need more. So I'm going to call P the number of variables. So biomarkers, say 23,000, that comes off of a CDF custom definition file for Affymetrix chip, for example, or a new generation compiler for RNA-seq. And in this 23,000 vertices, these genes that come out of this aggregation, we have up to 10 to the 5th edges. That's a lot of edges. And we might only have maybe 270 samples. That's the actual number of samples we ran in this challenge study I was talking about for the viral correlation between viral inoculants in the 2 categories that I described. So is 270 samples enough to be able to tease out all of these correlations? And the answer is no. If you restrict the correlations to be greater than, say, 0.8, 0.9, maybe you can ascribe a lot of confidence to those edges, right, that are really, really short, because those correspond to very high correlation. But the longer edges, they're very, very noisy. So this is basically the problem of big data, right? That there's a large number of unknowns, hubs, number of hubs being the number of genes, hubs being a gene that has lots of correlated co-expressors, if you're looking at gene expression, edges, or a number of subgraphs, which is exponential in P, the number of biomarkers. There's a very small number of samples to make inferences, and there's a crucial need to manage uncertainty. So let's just go back and recall what correlation is. I don't want to insult your intelligence, but it's good just to remember. Yeah, let's get this out. Sample correlation is basically a measure of collinearity, right? You have 2 genes, let's say, X and Y, in blue and green. The genes express over different samples according to this, call it a waveform or function over time. Let's say you're taking these samples over time. There's perfect correlation between these two genes. They're not exactly the same, but with a translation, that is subtraction of the sample mean, you can make them the same. And then the correlation coefficient will be equal to 1. If they're of opposite side, all you have to do is multiply one of these with a negative 1, and you're back to this situation, and that's a negative correlation equal to -1. Okay, so we're looking for sample correlations that somehow represent the population correlation. So there's a difference between sample correlation, which exists in your samples, and the population correlation that would exist if you had an arbitrarily large number of samples. You can never pay for that study, right? But there is some correlation there. And the question is, how accurate can we be in estimating that correlation from the sample correlation? So here's an example, which I hope will drive home the fallacies that can occur and that have occurred in lots of publications in scientific fields that we know about, where one has a list of genes, and let's just pull 2 of those out. And let's say this is the time index at which these samples were acquired. You have these blue and green genes again, and you're looking for correlation, and you say, okay, obviously they're not perfectly correlated either with -1 or +1 coefficients, but are they maybe correlated with correlation coefficient greater than 0.5? That might be biologically significant, right? It might be that you just don't have, this gene may have, these 2 genes may have a linear correlation, but there's some auxiliary variable you're not controlling for, right? If you could control for it, maybe it would be perfect correlation. So if you do the sample correlation over this 50 samples for these 2 genes, you end up with a number, and it's almost equal to 0. So you would say, no, they're not correlated at this level of 0.5 because the sample correlation is almost 0. However, if you want to be smart about it and say, well, actually, this is collected over time, and I'm doing time studies. I've given some treatment at time 0, and the organism that I'm pulling these samples from is somehow evolving over time. Well, maybe I should divide this up into windows. And you could ask, are there windows where I get 0.5 or greater correlation? And you can easily find windows for which that's the case. So here's one where we have negative correlation coefficient of 0.5. Here's one you have a positive correlation coefficient. So the answer to the question of are these correlated might be, well, yeah, if I have a window of time where things are changing, but you know, they come into phase and go out of phase 180 degrees, I'm seeing correlations. Well, these are completely spurious because those were generated from an uncorrelated IID source in MATLAB. Okay, so you can always find correlations if you look for them. Yes, exactly. That's the multiple testing problem. So not only have you reduced the number of samples, but now you've taken multiple windows. So the p-value associated with declaring significance at some at 0.5 correlation level is going to have to be corrected, right, in some way. And that's easy to do with 2. If you're looking for 1 correlation with 2 genes, it's easy to do. We know how to do Bonferroni corrections and bulk discovery rate analysis and so forth. When we go up to more sequences, we have a double whammy because now we have multiple testing not only over these time windows, but over multiple correlations. So with 20 correlations, you have, on the order of 200, 20 sequences, you have on the order of 200 correlations, right? 20 choose 2. And so now we can ask, are there patterns of correlation here? And again, at putting our threshold at level 0.5, we find 12 cross correlations. And as you might have guessed, this is completely random. I've just generated these from a MATLAB. right, generator. And we find these correlations, we find these patterns, and it's very easy to start imputing some mechanistic model for these spurious correlations. That we, unfortunately, then we see, graphs like this, where you have a network of correlations that are completely spurious, but they have survived this 0.5 level test. This is a real problem. Misreporting of correlations has led to retractions of published papers in journals like New Journal of Medicine, the other journals that have unfortunately not paid attention. The reviewers didn't pay attention to this problem of false positives. So this is a very interesting article by Young and Carr, who are director and co-director, respectively, of the National Institute for Statistical Science in Raleigh-Durham. And so they basically got the data from these various papers and tried to basically replicate the conclusions. And you can see that none of them replicated. None of these 12 studies replicated. In fact, 3 of them replicated in the negative direction. So it was the opposite conclusion that was actually the case when they replicated these experiments. So these are from the perspective of the credibility of the scientific enterprise, really big deals, right? If you can't trust papers that appear in the New England Journal of Medicine or the Journal of the American Medical Association, that's a big deal. Yeah. And so what's the problem here? Well, the problem is that there are always false discoveries, and there's a phase transition that describes where these false discoveries become a problem. So what does this show? This is a histogram, right? So what you see here is I've just drawn at random from 100 Gaussian random variables. I've taken 101 samples, and now I've computed all the correlations between them, sample correlations. I just plotted the histogram of those correlations. So you can see that there's these 100 variables, there's actually no correlation, so everything is spurious. Every one of these levels corresponds to a spurious correlation of, say, 50 correlations found at a level close to 0.3. And this point where we get a dramatic increase in the number of false positives, it occurs at some level of correlation, which depends upon how many samples you have. If you only have 10 samples, basically any correlation that you observe is going to be spurious, because the phase transition occurs very close to 1. So what this says is that we need to know something about where this phase transition occurs in order to be able to go in with our eyes open to right sample sizing a study where you have a large number of variables. These might be metabolites or some set of RT-PCR transcripts that you're testing. And the theory that we develop specifies exactly where these phase transitions occur under very weak assumptions. Basically, the assumptions are that everything isn't fully connected. These networks that I was showing you before, they're only on the order of p edges, not on the order of p squared, right? Which would be a totally connected network where everything was correlated with everything else. If you make that assumption, then you can get very accurate estimates of where these phase transitions occur. And now you can use these to say, well, if you only have 10 samples, you better be not be looking for correlations that are less than 0.89, right? Because they're just going to be spurious. If you have 25 samples, you can be pretty confident you find a correlation that's, say 0.8 or 0.9, because it's well above this critical threshold. So that's the principal point that I'm going to be making throughout this talk is the existence of these correlation thresholds and the importance of being able to specify them to be able to predict before you do your experiment, or once you've done the experiment, whether you have enough samples. All right, so the principles behind designing reliable correlation mining are basically to determine which among several regimes, sampling regimes you're in. These are mathematical limits, right? There's a mathematical statement. N is the number of samples, P is the number of variables, biomarkers you have. So the standard central limit theorem, law of large numbers, is the classical statistics that we learn, says that you've got to have N increasing faster than P, right? You got to have many more samples than you have variables in order to be able to compute p-values, to be able to use the central limit theorem to perform hypothesis testing with reliable uncertainty quantification. There's another regime which is we call a sample critical regime, which is when N and P both increase at kind of the same rate. So that constant C. So they both increase at the same rate. And for this, it turns out that, well, you can't say anything precise about the correlations, but you can actually say something about whether the correlations exist. You can't quantify the correlations, the value of the correlation, but you can ask, are there edges of a particular level? If this condition is satisfied. So this is the regime, if you like, where you can reconstruct the network, but you can't actually quantify how strong the edges are in the network. And then there's this other regime, which is we call the sample-starved regime, where the number of samples is fixed, and P goes to infinity. This is what's called the law of small numbers. So this is a much less familiar concept to people that have taken statistics classes, even advanced statistics classes. It's a new theory that's closely related to random matrix theory. Goes back 20 or 30 years to some work by Stein, Charles Stein. It's a central limit theorem, which is not to the Gaussian limit. It's to a different type of limit. And so you can let N be fixed and P go off to infinity. I argue, my premise here is that is the big data paradigm, right? That is where we need new statistical techniques in order to quantify uncertainty, is when we have an enormous number of variables, but we only have a few samples that are available to make decisions. And the N equals one patient. That's right. That's precisely right. So important to decide whatever procedure you're looking for the regime that you're in. This is just a table that gives some terminology, large sample classical asymptotics that's due to all these well-known names to people in statistics, going back to Sir Ronald Fisher. For small data, it has a great application domain. There's mixed asymptotics that have been recently developed in the 80s and 90s by some well-known living statisticians. And then there's this new regime which we are inventing and proposing, is the real big data regime that people have to be concerned about, which we call the purely high-dimensional regime. It's where the number of samples is fixed and the P, the biomarker dimension, gets arbitrarily large. All right, so why is correlation important? I've sort of given you one example for looking at correlation for two treatments, right? When you're trying to find pairs of biomarkers that are interacting, but there's much broader context. Of course, network modeling that I'm just talking about, learning or simulating descriptive models for the interaction, the co-expression, let's say, between genes. There's prediction, so forecasting a response variable, predicting whether somebody is going to recover from a pathogen response, or whether someone's going to get sick relative to say, a measurement of their interferon-inducible pathway before they show symptoms, of classification, and anomaly detection, localizing unusual activity. All of these involve correlation to some degree. And these are just equations that some of you who have worked in the bioinformatics statistical field will recognize. The linear minimum mean squared error predictor for doing prediction of y from x involves the inverse of the correlation of the independent variables, right, of the predictor variables, or the regressor variables, as they're called. For classification, if you're doing, say, quadratic discriminant analysis, and you're looking to discriminate between two different types of co-expression networks, one that has correlation sigma 0, the other one sigma 1, then it's the inverses, the difference in the inverse correlations that matter. And so this is the optimal way of testing that. And then anomaly detection, you have a Malone-Lewis test that can tell you whether, if you have some nominal co-expression that's in evidence in, say, some organism, is there a deviation at some particular time from that nominal due to some pathogenic insult or other environmental, say, or stress-related component. And that also depends upon the correlation. So this just is to emphasize that correlation just is not this kind of visualization tool that enables you to look at networks and graphs. That it comes in just about everywhere you have a problem of extracting some testable hypothesis from your data. And then there's, okay, so this just shows some of the categories of correlation estimation, graphical model selection, which is basically looking at where are the edges, right? The problem I mentioned before, where quantifying the value of the correlation is not as important as actually saying whether there is correlation or not above some level. Independence testing, correlation screening, all of these are different versions of the same problem involving estimating the correlation or the inverse of the correlation. Right? As I showed you before, the prediction, anomaly detection, and classification all involve estimating the actually inverse of the correlation, or equivalently the partial correlation, the normalized inverse. So you get a coefficient between -1 and +1 that characterizes the elements of the inverse correlation. That's called the partial correlation. And so this table shows the various regimes I was talking about in terms of how many samples you have relative to the number of variables and what you can do in those regimes. So if you have a number of, let's go to the standard regime where you're just trying to perform estimation, right? You know that there's a sparse number of correlations. So there's only on the order of p correlations in your model, not p squared. Then you're in this regime. And what this says is that n has got to basically increase as p log p, right? So n is increasing as p log p increases. So if you get more variables, you're going to have correspondingly superlinear more samples, right? In order to be able to do the parameter estimation, in order to minimize this norm squared error between, in this case, the inverse covariance matrices. Now, if you back off and you say, well, what if I don't have the money to buy p log p samples? What if I can only buy p? Well, then you can only do support detection. You can only estimate the graph. You can't actually put a number on the edges that connect different nodes in that graph. You can back off even further and say, well, I don't even have, I mean, p might be on the order of 23,000, and purchasing 23,000 RNA-seq chips is pretty large undertaking. So maybe you only have log p, order log p samples. In that case, you can't even localize where those edges are, but you can certainly say whether there are edges or not. Right? So if you take some subnetwork, some subset of the biomarkers. If you only have on the order of log P samples, you can reliably determine whether you have edges. NE is the number of edges in that subnetwork being greater than 0. And if you back off even further, if you have fewer than log P samples, you can still do something. You can discover those. You can discover that there are edges for correlations that are very close to 1. Right? Here you can discover edges for correlations that are close to rho star, which is this critical phase transition threshold. Here, this critical phase transition threshold goes to 1. So all of these regimes are important. This regime is the most punishing. This is how do you estimate the performance of your estimator, right? So let's say you have an estimator of correlation. Now you want to build a confidence interval around that estimator. Well, now in order to build the confidence interval reliably, if you don't know the underlying distribution, we're not assuming Gaussian distribution or anything here, then you need on the order of exponential in P samples in order to make a good estimate of the performance. Yeah. Well, that's a good question. So P, yeah. The question is, what if you're not sure of what the value of P is? So that can arise before you do the experiment, right? So you say have a choice between assaying, a relatively modest number of biomarkers versus a very, very large number of biomarkers. And there you have to basically choose, right? What do you want to, what kind of a budget do you have for biomarkers versus your budget for samples? Now, in a setting where you're doing data analytics, you know P. Because P that's the sensor, that's the microarray, that's the RNA-seq number of probes, right? So you generally know P after you've done the experiment, right? But in designing your experiment, what you're pointing out is very very important. And in fact, the example I'm going to present at the end gets at a part of that question. Yeah, another question. Yes. Yes. Exactly. And so that's the basic picture here. Okay. So let's say that you're collecting samples, and this is an argument for adaptive sampling, adaptive data collection. You don't have $1 million to collect 23,000 samples, right? But what you can do is as you start collecting samples, you first enter this screening regime, right? Where you can detect whether or not there are edges, right, among your variables. And so once you enter this regime, then you know that this experiment has legs, right? There are differences, let's say, between the co-regulation network under treatment A and co-regulation network under condition B, but you don't know you don't know how to localize those differences. So then you take more data that's with the same treatment until you have enough data that you enter this other regime where you can do support recovery, that is network estimation. So you can find out that there are edges and you can actually localize where those edges are. And now if you want to build confidence intervals on the values of those edges, then the first thing that happens is you enter the estimation regime, where now you can focus your collection just on those edges that you found, right? All the other variables you can throw away. So you go from 23,000 variables at the screening stage. Support discovery, maybe that falls down to maybe 150 variables. Now you can spend a lot. You can spend a lot more time, or you can spend your limited resources and a lot more samples of fewer variables and enter this estimation regime. And then in the future, if you want to put confidence intervals, you get into the performance estimation regime. So that's the progressive correlation mining paradigm that comes directly out of this type of point of view, that with different amounts of data, you can predict, knowing what the value of P is and knowing how significant the correlations are that you want to discover, design the experiment so that you're collecting enough samples. Yeah. No. No. Oh, no, definitely not. Yeah. The only assumption, let me just be clear, the only assumption here is that the network of correlations is sparse. Right. Yeah. So that, everything's not connected to everything else. Yeah. Right. If you have prior, so if you have prior information that tells you that, because there's been study A, B, C that have established that there's no evidence that there are links between this pathway and that pathway, well, then maybe you don't look at this pathway, right? That helps you to reduce the number of variables. But, you don't always believe what you read. So you may want to, do just some initial exploratory design where you use the full genome, let's say, and verify, right, what you've read. Yeah. So this allows you to go back and forth between these regimes. So I'm not going to spend much time here. I'm running out of time because I wanted to get the main ideas across on the mathematics. But there's hub screening is just a version of edge screening where you're looking for, hub screening is a version of screening for vertices that have edges, but their degree of those edges has to be greater than or equal to some quantity. So for example, you might be looking for nodes that have degree, greater than 10. This would be one of them here. So you can screen for nodes, for biomarkers that are connected at some level of correlation to more than 10 other biomarkers, right? That's hub screening. And hub screening of order 1 is simply looking for connectivity, right? Being a vertex with at least one edge is the same as being a vertex that's connected. So one can do this, one can formulate the problem of hub screening through a theorem. I'm not going to dwell on this theorem at all. I'm just going to point out that using this theorem, you can theoretically define where these phase transition thresholds occur for various numbers of samples. And in this particular case, we have 500 biomarkers, 500 genes, if you like. We get a mathematical equation for the phase transition point where things start to go to pot. And you can look at false discovery probabilities so that this theorem gives you what looks like almost a Poisson limit of the number of false discoveries, where the rate in that Poisson limit is precisely determined by this phase transition. And then you can see that this critical threshold depends on the hub degree that you get mathematically. You can look at this as a design matrix, if you like. So these curves tell you for different numbers of variables. So let's start with 10,000 biomarkers that were in these curves here. This is looking for hubs of degree 3 or more. This is just looking for connectivity. The number of observations you need versus the critical threshold. So if you want, for example, to discover correlations that are greater than 0.7 in magnitude, then if you want to discover those correlations in terms of just connectivity, right, not hub degrees of greater than 2 or 3, then you're going to need about 50 samples, 50, 60 samples in order to hit that if you have 10,000 biomarkers. If you go to 10 billion biomarkers, you only have to basically double the number of samples. in order to retain the same correlation sensitivity. This is the blessing of big data, right? It's that you can accommodate enormous increases in the number of biomarkers with only a small increase in the number of samples because of this logarithmic dependency between the sample versus number P of, we have this log P over N regime that I was talking about before. So samples buy you a lot if you're in this sample-starved regime. Now, if you're in the sample-rich regime out here, they don't buy you a whole lot because we're getting pretty flat, right? This is just showing some actual results. So this is the respiratory virus challenge study I was mentioning before. This appeared in several papers, most recently in a Science Translational Methods, a paper with Amy Zass. So inoculation is given at this point 0. They're pre-challenge samples. Red and green are subjects that started to become symptomatically ill at some point during the challenge study. This is time here. So we've divided the post hoc, the subjects that were asymptomatic and those that were symptomatic into 2 groups. We had an attack rate of approximately 50% in this study. And so these are gene chips that were collected. This was in previous generation technology because we did this experiment in 2008. This just shows you the data. We did it for respiratory syncytial virus, H3N2, H1N1 flu, also the common cold, various locations, retro screen in London, University of Virginia, total of over 2,000 samples, which I'm not showing here. These are the duration of the studies, and over 120 subjects. So it's a pretty massive undertaking, a challenge study that produced a wealth of data that I'm going to show you just snippets of results. The papers you can see more detail. So this is for the particular H3N2 study. In fact, the first one we did, D2 and D3, were 2 H3N2 studies. D3 was a repetition. 17 subjects were inoculated, sampled over 7 days, leading to this heat map of each one of these is a gene chip with number of samples. I've partitioned this into the clinically determined asymptomatic and symptomatic categories post onset, pre-onset of symptom, and we used the brain array, the Michigan brain array custom definite. You built it quite a while ago, and lots of people are using. Yeah. And we had symptoms that were scored 0, 1, 2, 3 on a 10 symptom scale, including things like runny nose and fever and coughing and so forth. And so now you look at the samples, these categories that I showed you before. We have pre-inoculation samples and post-inoculation samples. The post-inoculation samples are symptomatic or asymptomatic classes. We know the number of samples. We know the number of biomarkers. So we can compute these phase transition thresholds. So in this regime, the pre-inoculation samples, the critical threshold is 0.7. So anything you find that's less than 0.7 in sample correlation is completely unreliable. And if you want a 10 to the minus 6 family-wise error rate threshold, then you have to raise that to 0.92. Right? Yeah. And so I'm only going to focus on the pre-inoculation samples just to give you a taste of what we can do. So this is now screening the partial correlation. This is the inverse correlation, as opposed to the correlation itself. And we compute the inverse correlation through a pseudo-inverse, so what's called a Moore-Penrose generalized inverse, because the inverse doesn't exist. We only have a few samples, right? We only have effectively 34 samples, but 12,000 biomarkers. So there's no inverse to this matrix. But the pseudo-inverse can be computed, and the theory we developed applies to the pseudo-inverse also under these assumptions that I was mentioning, sparse correlation. And what you can see is very interesting. This is for the pre-inoculation samples. So these people are coming off the street. There's no treatment yet. Coming off the street, this is measuring what is their interactome, right? And so what you see here are some Affymetrix genes that are basically controlling housekeeping in the Affymetrix human 133 gene chip. And then we have some nonspecific ribosomal protein transcription factors here. And now here, this is very interesting. These are part of the interferon-inducible transcription factor pathway that basically controls the immune response system. So toll-like receptors, nod-like receptors are in this pathway. And this is just saying that their pathways are working, right? The sentinels are there. These individuals have they have working immune systems. And so this result, if you looked at just the correlation, you find a mess of connections and not modules that one cannot make any sense of. But it's through the inverse correlation that you observe this very nice picture that really reveals the inner workings of the theory. And then you can also draw pictures like this, which show as a function of the correlation that you found, and here I've set the correlation threshold to 0.92 for false positive rate of 10 to the minus 5. And so this would be for connectivity. This is the degree of the node. This is degree greater than 1. So this would be connected to at least 2 other genes, 3 other genes, and so forth. And at this threshold, we found biomarkers that were connected at 0.92 or greater to, at most, 14 other genes, co-expressed with 14 other genes. And then you can look at the various genes that fall along these categories and sort of pick them off in this visualization of this 12,000 node network with many connections. And I'm going to finish now with this predictor design example, where instead of looking for a co-expression between genes, you're looking at the correlation or the association between gene expression on these 12,000 genes and some other variable, some other biomarker, which here is going to be symptoms. So we're going to try to effectively find those genes, not just that are correlated together, but that are highly predictive of symptom. This is a symptom in the future, right? So the concept here, this DARPA funded this study. The concept was to basically cure before. That was the byword, right? Cure before symptoms occur. Yeah, right. Typical DARPA speak. And it, but the idea, certainly is very interesting that you can try to predict ahead whether symptoms will occur. Here's the symptom maps as a function of time for all the different samples organized between the symptomatics. You can see that a lot of the asymptomatics were complaining of symptoms too. So this is a difficult problem, right? Yeah. The asymptomatics were, there were some in here which were psychosomatically hyper people. I must say also in here, I'm not showing this data, but we also have viral titration data for shedding. There are a lot of typhoid Marys in this group. So, So it's a difficult problem. We're relating self-reported symptoms to molecular expression. So don't expect to see, numbers that'll throw you off your seat here. Right? The numbers are not going to be as impressive as one might expect if you were looking at correlating something that was more tangibly related to co-expression. But my point here is to show that one can do significantly better using this progressive sampling strategy that was motivated by these regimes of operation. So this shows why it makes sense to do progressive sampling. Let's say that you're doing experiments that are very costly. We paid about $250,000 for each challenge study. The number of probes per chip In terms of pricing, if you looked at adjuvant customer raises back a couple of years ago, actually now. Oh, no, February 25th. Yeah, so a couple of years ago. So it's basically linear, right? So what this means is you can pay a lot for a lot of probes and maybe a few samples, and then you can back off and get a lot more samples with fewer probes once you've discovered where the edges are. That's what this, yeah, that's what this picture is made to communicate. And so this just shows what we're trying to do, trying to predict these symptoms with the samples of molecular level gene expression, and the predictive correlation screening, where you knock down the number of genes that you're going to assay as a function of the correlations that were observed with relatively fewer samples, but lots of biomarkers that you weren't able to quantify, but you're able to detect. And so this is related to a lot of other approaches, compressive sensing, sparse regression, and so forth, that I'm not going to dwell on. There's a theorem that comes with this that tells you how you should sample. This theorem in particular is very important. You have a total budget of T dollars. This is the number of samples you should allocate to get optimal detection performance, optimal prediction performance at the end of the day, after you do this two-stage process. Screen, with large numbers of biomarkers, a few samples, then use the screened samples, which are much fewer, and purchase a lot more samples of those in the future. And so N is the number of samples that you should buy that have the full biomarker set. And it's only log of the total number of samples. So this regime tells you that, again, you have a blessing of dimensionality that you only need logarithmically to allocate logarithmically in the number of total samples to the expensive one. The rest can be cheap. So you can take more samples, but overall you've sampled fewer biomarkers on the average. And this just shows the advantages. So this is our two-stage procedure as a function of N, the number of samples, where you drop from 12,000 or 10,000 here to a fraction of that. And as compared to state-of-the-art methods, which are very far away from where our operating point, right? In terms of average prediction, root mean squared error, we're also doing quite a bit better. The computation is much less than it is, for doing these other types of methods, which involve, lasso involves optimization, linear programming. The method that we propose, it's basically thresholding with a pseudo-inverse. We applied this to the symptom prediction problem. And again, these numbers aren't great in the absolute sense, 0.6953 in terms of mean squared prediction error. It means that standard deviation is, there are integer values, right? The 0, 1, 2, 3, 4 are the different symptom levels. So what we're saying is that, you can predict within maybe 2 levels, right, with this type of standard deviation. But the important thing is that when you use this new method, you reduce that by about 15–20%. So you reduce the error by 15 to 20% simply because we're able to sample smartly and not sample in batches and devote our resources to sampling biomarkers that are not predictive. Right? We find out which ones are predictive first, and then we sample. And then these are just some of the genes that came out, which isn't important. This is the conclusion. So when requires lots of care in this big data setting where you have many fewer samples than biomarkers, N is much less than P, inadequate to apply the standard central limit theorem type of measures. Bootstrapping is very unreliable. You will, no matter how much bootstrapping you do, if you have insufficient sample size, you're always going to find spurious correlations and associations. Bootstrapping is completely data-driven, so the data is not sufficient to check itself, right? You need to have a mathematical theory to tell you just what are those critical phase transitions as a function of the number of biomarkers and the number of samples. So we have this new big data setting, purely high dimensional with universal phase transition thresholds. And then I haven't talked about these other topics of structured covariance. So these are forms of prior information that one can incorporate. Toplets is an interesting one because toplets means that you are perhaps correlated over time, but you're stationary over time. Yeah, things aren't, the correlation isn't changing over time, right? So toplet structures can be very useful when you're doing longitudinal serially sampled studies with correlations from time to time and also correlations between biomarkers. And then one can also extend this to nonlinear correlations using a method we developed called measure-transformed correlation mining and spectral correlation mining for stationary time series, again, relating directly to this toplet structure. So that's basically the story I wanted to tell you today, and I thank you for listening. Yeah. Yes. Yeah, that's a good question. So the purpose of this study was not so much to put together a tool as it was to demonstrate the advantage of doing this kind of aggressive sample sizing, right? But we are indeed concerned about how to apply this in a kind of setting where you're trying to do prediction of symptoms. And so what we've done, and I haven't showed the data here, is that what I showed you in this example was separate networks for each symptom, right? That was averaged over all symptoms. What we've done is we have also developed a network that best describes all symptoms, right? So that network is penalized or scored, if you like, on the basis of how well it discriminates not between the levels of a given symptom, but the levels of several symptoms. The problem here is that with this panel of symptoms, there are 10 symptoms. They're the standard panel, clinical panel, that is used to compute Jackson scores for respiratory viruses. But some of these viruses don't, for example, cause a febrile illness, right? So fever is very difficult to predict, right, in those situations, because any report of fever, feeling feverish, is probably going to be highly unreliable because it's self-reported. So one has to specify which symptoms are the most relevant for the particular clinical objective that you have in mind, and that's up to the clinicians to describe. But very good question. Thank you. Exactly. Yeah. Yeah. They're very descriptive, and there are people that like that are hypochondriac, and there are people that they see a needle and they start to get symptoms, right? And here they're not seeing a needle. What they're seeing is a cotton swab with H3N2 virus on the tip of it. So, yeah.
So you show that a baseline is lots of stuff that can correlate with biological correlate. The question is your other interactions, like one of those things connects to a number of different things, but that correlation is so much stronger for the correlated one, that you start losing the ability to find these deeper correlations because mathematically during
Yeah.
that. How do you collapse that network into one thing and call it F, and then use that to find the other correlation?
Yeah, great question. So if you use correlations, and I didn't emphasize this enough, you use the standard correlations, you get into this problem, this masking problem. You know, if A is correlated to B and B is correlated to C, then they're all correlated, right? On the other hand, if you look at the inverse correlation, partial correlation, then A correlated to B correlated to C, the partial correlation between C given B and A only depends on B. Right? So effectively, that's why I didn't show you the other network, which was a complete mess. This network is the partial correlation network, which eliminates some of those problems, those masking problems. Not all, but it does eliminate some of them. And then what one can do is, as you suggest, you can design the threshold so it's low enough so that you discover a little more than what you really would be comfortable with, but you still have dropped down the total number of biomarkers you have to deal with by maybe a factor 10. And then you can focus into those and do a subsequent analysis. Yeah. 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 So you're talking about now having a time series of, maybe treatments or, exactly. Not the other. Yeah, and those are all, great open problems. Let me put it that way. And we're following up on them. We have a great data set that's longitudinal and serial sampled, but we don't really have enough samples to look at the kinds of questions you're talking about. So we still have to aggregate, over large chunks of time in order to get the number of samples that are needed. So, yeah.
So our key distinction was N smaller, probably much smaller than P.
Yeah.
We usually describe this as overfitting.
Exactly.
And this problem is highlighted in the report from the National Academy of Sciences on predictive omics-based tests called the evolution of translational omics.
Yeah, I've seen that.
But what I wanted to ask you is, how much closer to P does N have to be to make some of these analyses feasible?
Yeah, and that really gets to this table I was showing. Yeah, let me go back to it just to put a point on it. Yeah. Yeah, and this really tells you what you need. Yeah, this is the story. I could have given the whole talk just with this one slide. Right? So if you look at these regimes, forget about all these other. Just look at this one row. This tells you, if your task is really to estimate correlations, and you don't want to overfit, then you need to have basically N more than P. It's got to be more than P. And maybe that P is a P of a set of biomarkers that you've discovered using screening, support detection, or detection from a much larger set of biomarkers. But still, N has got to be, on the order of P log P in order to be able to do that reliably. On the other hand, if you don't have that number of samples, you have to figure out which one of these regimes am I in, right? Am I here? If I'm here, where I only have log P of samples, well, I can't do network discovery, report detection, finding the edges. I can't certainly estimate all the correlation coefficients. I certainly can't do, ascription of uncertainty to those correlation coefficient estimates. But what I can do here is I can detect if there are changes, right, between treatment A and treatment B in terms of the sample correlation networks that they describe. So by using this type of table, you can put yourself into the right task. I mean, we usually don't think about that in this setting. I mean, we usually think, well, I want to test these hypotheses, and these hypotheses involve estimating the correlations with this level of confidence. And now I do my sample sizing, and I find out, well, I can't afford it. Right? What this tells you is take a step back, see what you can afford, and then this table will tell you what you can do. Right? And then you can adapt. Right? Yeah.
Yeah. Hi. For the phase transition threshold,
Yeah.
what do you mean exactly? What's the meaning of the phase transition threshold, and how do you find it? The other question is, does it depend on the underlying distribution?
Yeah, very good question. So the phase transition threshold is the threshold of correlation coefficient that is the necessary and sufficient threshold for you to be able to reliably say that you're on the right side of this bump, right?
But what do you mean exactly reliably? Does that
Reliably
mean corresponding to a p-value?
Yes.
Or?
So yes. So the p-value I didn't show you. It was in this theorem here. If I could find, well, I wouldn't bother showing it. But there's associated with this phase transition threshold, which roughly speaking corresponds to a p-value pretty close to 1, 0.95 or something like that. If you specify a p-value, you're going to get another threshold, which you specify a threshold on your family-wise error rate, and you're going to get a p-value threshold, right? And that'll be larger than this. That's why in that example I was showing before, where we were looking at the baseline samples, the phase transition threshold was at 0.7, right? But if you wanted a p-value that was less than 10 to the minus fifth, well, then the threshold is 0.92, right? So the p-value is not shown explicitly in this picture, but it comes right out of the theory. And now, right, your next question is in what sense does it depend upon the underlying assumptions? So this, there's a, the short answer is, as long as your samples come from a distribution that is elliptically contoured. So it doesn't have multiple bumps. It does have multiple bumps. You're going to have to cluster and focus in on one of the subclasses that has a elliptically contoured distribution. As long as that ellipse that gives you the constant contours of your distribution isn't too flat, meaning that there are collinearities, right, in your data, meaning that there are correlations that are almost equal to 1 in your data, then you're going to get away with this. And so what we're saying is you can put down sparsity coefficient. You can state that you have elliptically contoured distributions. And at that point, these will be reliable. Yeah, thank you. Yeah, thanks.